Okay, I think.
Awesome. All right. So thanks a lot to everyone. Thanks a lot for attending this month's NeuroMeetup talk, for which the title today is going to be Generative Adversarial Networks for Applications in Neuroscience. And to discuss this topic, we have the great pleasure to have as our speaker today, Purnima Ramesh. So Purnima studied physics for her bachelor's and master's degree at the Indian Institute of Science, Education and Research in Kolkata. And she's now working on a PhD with Professor Jakob Macke in the Machine Learning in Science group of the University of Tübingen in Germany. And namely, she's applying GANs, generative adversarial networks, to problems in simulation-based inference in neuroscience. And so that being said, Purnima, the floor is yours.
Yeah, thank you so much, Camille. That was a very nice, very kind introduction. As Camille said, I'm going to be talking about applications of generative adversarial networks for neuroscience. I will not be talking about simulation-based inference today, but two other projects that I am working on in my PhD, and this is work that I've done in the machine learning for science group at the University of Tübingen with Professor Jakob Macke. Please feel free to jump in with questions, but I can't see the chat, so just go ahead and interrupt.
Yes, regarding the question, if you have any question I'm addressing to the audience, please write them down in the chat, and I will transmit them to the speaker, either during the presentation, if we have the opportunity, or afterwards. We will have this question and a Q&A session after the talk.
Cool, thanks a lot. All right, so stepping back a little bit, we can think of a neural network as a flexible function approximator. And what I mean by this is the neural network basically takes in some input and it spits out some output. And this mapping from the input to the output is as flexible as you would like it to be. And typically what the output that comes out of the network, we want it to match some target. And the way we train the model to match this target is using some loss function. And the loss function encodes a distance metric between whatever comes out of the network and whatever target we have. So this could be mean squared error, a likelihood, a KL divergence. All of these different kinds of loss functions should encode some notion of distance between the network outputs and your target. But sometimes it can happen that we can't define a loss function. So if you had image data, for example, or if you had audio files, then it's very hard to come up with a loss function that precisely tells you how far apart two images are or two audio files are. But if you still want to train a network that would produce images for you or produce audio files for you, then what do you do? And this is precisely the kind of situation in which generative adversarial networks, or GANs, are really good. So what happens with a GAN is that you simply replace your loss function with a second neural network, because as we know, neural networks are good function approximators. And so you use a network to approximate also the loss function. And so how GANs work is now you have a generator network, which is the network that that is your input output function. And what it does is it takes some input and it produces some output that you want to match to the target. And then what the discriminator does is it takes the output of the generator and it classifies it as fake. And it takes the targets that you want to match and classifies them as real. And the adversarial bit comes in because you basically train these networks against each other. And what this means is that you have a single loss function, which in the vanilla GAN case is a cross entropy loss. And you minimize this function with respect to the generator and you maximize it with respect to the discriminator. So basically you have adversarial objectives. You're trying to push the loss function down for the generator and you're trying to push it up for the discriminator. And what this means is that the discriminator is trained to get better and better at classifying real and fake data. The generator is trained to get better and better at fooling the discriminator into saying that whatever it produces is real. And this is how you achieve it with the cross entropy loss. And what happens at convergence, so when you finish training everything, is that the generator has learned an input-output function, and the discriminator has learned a loss for the generator. And because we have this adversarial setting where each network is trying to beat the other, at convergence, what you expect would have happened is that the generator has learned to produce outputs that look exactly like your target data set. And this is essentially the setup of a GAN. And what I'm going to talk about now is basically GANs are extremely good models for high dimensional data sets, precisely because you're only working with samples, and therefore you don't have to define loss functions. So some of the most famous GANs are for images. Sorry about this. So there's the pix-to-pix GAN, which basically converts, it does image-to-image translation. So if you give it black and white images, it's going to like translate them into color images, or if you give it a sketch, then it's going to color them in. Another very famous GAN is called style GAN, which produces images of people who do not exist. There's another GAN called stack GAN, which basically translates text to images. And all of these are GANs that deal with high dimensional data in the form of images. And in none of these cases is it easy to encode a loss function for the things that you want the network to produce, but nevertheless, you can train them to produce these outputs. And I'm going to talk about applying GAN specifically to data in neuroscience, taking advantage of these properties where you can deal with high dimensional data without having to specify a loss function for it. And the first application that I'm going to talk about is reproducing neural population activity. And this is work that I did with Mohammed Atai and Yaqub, my supervisor. So what exactly do we mean by reproducing neural data? So we know that neural population activity in the brain is driven by signal and noise. And what we mean by this is that the population activity is affected, of course, by some input stimulus. But there's also some latent process that we don't observe that also contributes to the activity that we measure. And what it manifests as is this. Suppose we have two neurons whose activity that we've recorded in response to two different stimuli. So the two colors, red and black, indicate two different stimuli. And each point in this plot represents one observation of the activity for neuron 1 and neuron 2. And this first plot here shows us that the activity is positively correlated with the stimulus. And what this means is that the activity of neuron 1 increases with the stimulus. So the activity of both neurons increase with the stimulus, but the activity of the two neurons themselves aren't correlated. And what I mean by this is that the activity of both of these neurons independently increase if I increase the strength of the stimulus, but there's no other structure in the responses. But if you look at the plot below, then what you see is that the responses of neuron 1 and neuron 2 do increase when you increase the strength of the stimulus. But additionally, there's this structure in the responses where they're distributed more narrowly within a particular stimulus, and this correlation between the activity of neuron 1 and neuron 2 is because of some latent process that we have not observed, some latent noise process. And essentially, when we say we want to capture or when we want to reproduce neural data, then we want to reproduce both of these features. We want to capture both the fact that neural activity is driven by the stimulus and is therefore somehow correlated with it, but also that the activity of the neurons is correlated with each other because of some noisy latent process. concretely, what this means when we want to reproduce these features is that if we have some measured or observed spike trains from some region of the brain, then we want to set up a model that generates spike trains such that the mean and the variance of these population spike trains match. But we also want to match the noise correlations. So whatever correlation there is in the population activity because of your latent noise process. And additionally, we also want to match any higher order statistics, which are encoded by something called a population spike histogram. So we want to match, we also want to capture relationships where, for example, neuron A is correlated with the activity of neuron B, which is correlated with the activity of neuron C. And there's this kind of triplet relationship, which we also want to capture. And if you want a model to reproduce all of these features, then we are going to have to define a loss function that encodes all of these features. And this is something that is typically quite difficult to do. You do have lots of models that allow you to capture the mean and the variance, and more rarely some that also allow you to capture pairwise correlations. But capturing more than this is hard because it's hard to define a loss function. And as I set up in my introduction, this is precisely the kind of situation in which GANs can help. And so what it means to use a GAN for reproducing neural data is basically you have some measured activity in response to stimulus for some brain area, and you want to set up a generator network, which also, given the stimulus, produces population activity. And then you want to have a discriminator network, which classifies your experimentally measured spike trains as real, and the spike trains that come out of your generator network is fake. You train the generator and the discriminator adversarially, keep training, and at convergence, what you should have is a generator that produces population spike trains that look exactly like the measured spike trains. And this is nice because the GANs are very flexible with respect to the dimensionality of the data set. So if you have an arbitrarily large population of neurons, this is a setup that would still work. Two things that I quickly want to note is that one of the challenging things about using GANs for reproducing population spike trains was that we needed to deal with the discrete nature of the spike. So because of the fact that you have ones and zeros, it's very hard to estimate gradients to train the network. And this is one of the things that we had to deal with. And I'm happy to talk about it in more detail if it comes up during questions, but I will not get into it here. And then what we did was, once we figured out how to train the GANs with spikes, we fit them to spike trains recorded from the macaque V1 from an open data set that's available on CNS. And then what we wanted to do is we wanted to check whether fitting these GANs is better than what models are already available to fit spike trains. So the first model that we compared it against is a supervised model, and this is basically just your generator network, your neural network, which is producing your spike trains, and training it with a loss function. You have a well-defined loss function, and this is basically what we use to train it. And this model captures the mean and the variance of the population activity. And then the second model that we use is called a dichotomized Gaussian, or the DG model. And I don't want to get into the details of the model, but the thing that it's important to note is that this is a model that captures the mean, the variance, and the pairwise correlations in the population activity, but not the higher order correlations. So these are the two models that we compared our GANs against while we fit to the macaque V1 data set. And so we train our models, and then we generate data from it in response to the input stimulus. And this, what I show here in the first plot, is the real recorded data. And then this is the data that we generate from the GAN and from the supervised model and from the DG model. And just looking at it by eye, it seems that all three of them seem to reproduce exactly what the real data looks like. So we broke it down into some more concrete metrics. So here I am plotting on the x-axis the mean firing rate of the neurons in the real data, and on the y-axis I'm plotting the mean firing rate from each of the model generated data sets. And as you can see here, all three models, so the GAN in blue, the supervised model in orange, and the DG model in green, all three of them capture the mean firing rate perfectly. But when I now plot the pairwise correlations, and here this is the total pairwise correlation, so this is both correlations because of the signal and because of a latent noise process, we find that the GAN in blue and the DG model managed to capture pairwise correlations, but the supervised model doesn't manage to capture it exactly. And breaking this down a little bit further, we find that this is because the supervised model just does not capture the underlying latent noise process. It only captures the response of the neurons to the input signal, whereas the DG model and the GAN capture both. More interestingly, when we plot the population spike histogram, and this is basically a plot that shows you higher order correlation. So if you had neuron A correlated with neuron B, correlated with neuron C, so these kinds of like triplet correlations or higher order correlations with 4 or 5 neurons, then the population spike histogram captures this. And this line in black here is the ground truth population spike histogram. And what we find is that the GAN captures it, the GAN in blue captures it almost exactly, but both the DG model and the supervised model fail. So essentially, cutting a very long story short, what we find is that the GAN manages to capture more features of the data than either the supervised or the DG model. So this is clearly a point in the advantage of GANs. However, something that I probably should have mentioned at the start of the slide is that this is all on the training data and not on the test data. And when we actually tried to check whether the GANs generalize well to test data, what we find is that they don't generalize at all. So here I'm plotting again the mean firing rate, the pairwise correlations in the population spike histogram, and the GAN seems to do fairly well here. But then when we look at the peristimulus time histogram, or the PSTH, where I'm plotting the mean firing rate at each time point, then we find that here, where the black indicates the ground truth data, the GAN just doesn't manage to capture it at all in blue. But surprisingly, neither does the supervised models, even though it's explicitly fit to capture the mean firing rate. Neither of these models managed to generalize the test data. And so when we looked into it a little bit more, we realized that this was not a problem with the models themselves, but more a problem of the data set, where the signal to noise ratio wasn't high enough for the models to pick up on the stimulus driven, to at least to properly pick up on the stimulus driven aspect of the population activity, and therefore overfit to the training data and didn't generalize the test. So then what we did was, when we artificially simulated data with a higher signal to noise ratio, here we find that the GANs did generalize really well. And additionally, what we also did was we simulated the data such that the latent noise process was heteroscedastic. And what this means is that the strength of the noise is also dependent on the stimulus. So it scales with the value of the stimulus input. And this is a setting which neither the supervised model nor the DG model can cope with, but clearly a setting in which the GANs can. So one thing that we can ask is, what advantage do the GANs confer? Why does it matter? Why should we bother fitting GANs that capture all of the features of the population activity? Why is it not enough just to have a model that fits the mean and the variance of the population activity. And to answer this, what we did was we took the stimulus that was presented to the monkeys from which we had our ground truth recordings, and the monkeys were basically watching a video consisting of these frames that I'm showing here. And then what we asked was, which of the pixels in these frames strongly drive the activity that the GANs produce and strongly drive the activity that the supervised model produces. So essentially we're asking which pixels in these frames contribute to the output of the GAN model and contribute to the output of the supervised model. So here, what I've done is I've superimposed a sort of superimposed a feature importance heat map on top of the frames to show you which pixels strongly drive the activity that the GAN produces. And here, the white pixels are basically those pixels that strongly drive the activity. And what we can see is that these pixels tend to be concentrated around the face and the hands of the monkey in the video across all of these frames. And what we can also see qualitatively, at least, is that there isn't a center bias. So all of the pixels aren't simply concentrated in the center of the frames. And additionally, what we find is that if we did the exact same thing for the supervised model, then the features of or the pixels of the stimulus frames that strongly drive the activity are completely different. And remember, the supervised model captures only the mean and the variance of the population activity, whereas the GAN captures a ton of other features. And what we can conclude from this analysis is basically that if you had a model that captured all of the features or more features of the population activity than just the mean and the variance, then our understanding of which stimulus features strongly drive the activity can be completely different compared to if you just used a supervised model. And therefore, whatever inferences we want to make about the underlying brain area, so in this case, the primary visual cortex, whatever inferences we want to make about how visual cortex functions with respect to responding to different stimulus features can be completely different depending on which aspects of the population activity you capture. And therefore, it's important to think about whether or not it is useful to capture all features of the neural population activity when reasoning about which stimulus features are important to the neurons in this particular brain region. And this is precisely why you would want to think about using GANs as a generative model of population activity. So to summarize this part of the talk, we basically aim to capture both signal and noise in neural data. And we use GANs to try and accomplish this. We found that the GANs outperformed the supervised and DG models in terms of capturing different features of the V1 population activity. And we also found that the capturing correlational structure in the activity shows that it could be driven by different stimulus features compared to capturing just the mean and the variance. And therefore, it's important to think about whether it's useful to capture all of this additional structure in the neural population if we want to reason about the stimulus features that drive population activity. I want to pause at this point to take some questions because the next part of the talk will be about a different project. And in case anyone wants to ask questions about this part, then I think this would be a good point to pause.
So far, we don't have any questions in the chat. But if I may, I have a question actually. So the plot you've been showing, in which you have been comparing the different performance of the GANs of different methods.
Mm-hmm. This one?
Yes.
Mm-hmm.
Yes. Well, actually, when you were showing that that GANs were not generalizing very well to test data,
mm-hmm.
to be like some kind of telltale sign of overfitting, did you just simply try to play around with the structure of the GANs of the neural networks that you were using? For instance, just trying to use smaller networks to try to avoid overfitting. Did it make a change or not really?
Yes and no. So fitting GANs with smaller architectures and sort of like doing so that kind of like tuning in terms of architecture and hyperparameters was definitely something that we tried. But unfortunately, we had networks that would overfit a little bit less and overfit a little bit more, but none of them that really completely generalized to test data. So this is something that we did try. And we found that it didn't really help with the generalization. What really did help was increasing the signal to noise ratio of the ground truth data. This is one thing.
Okay, cool. Yeah, I think you're back. I think we had a small connection issue.
Okay.
So really sorry about that. Can you just redo your re-answer the question? Because
Okay.
you're done right at the beginning of your answer.
Okay, so basically what I was saying was that we did try lots of different architectures and we found that some of them overfit a little bit less and some of them overfit a little bit more, but none of them fully generalized to test data. So, okay. we did really establish that the problem was the signal to noise ratio. This is one thing. And additionally, I do want to mention that hyperparameter tuning for GANs in terms of architectures is quite difficult. And this is something that I will talk about towards the end of the talk, but yeah, it's typically quite difficult to do in any case. So I can't definitively say that there doesn't exist an architecture or hyperparameter setting that wouldn't generalize the test data. Yeah,
makes sense completely. Thanks a lot. We still don't have any question in the chat, so to the audience, please feel free to ask any question you might have, and otherwise, Purnima, you are free to keep going.
Okay.
Thanks a lot.
All right, so I'm guessing there aren't any more questions, so I'm going to move on to a different topic. So this is the second application of GANs that I'm currently working on, and this is very much this is a project that's very much in progress. So it would be nice also to just have feedback and a little bit more discussion about what makes sense and what doesn't. So I'm going to be talking about meta-learning synaptic plasticity rules using GANs, and this is work that I'm doing in collaboration with Tim Vogels and his PhD student Basil, and also with Jakob, who's my supervisor. So the idea is that we think synaptic plasticity is important for learning, for memory consolidation, for development in the brain. And we have lots of hypotheses about what mechanisms there could be for synaptic plasticity. So we know of Hebbian plasticity, we know of the BCM model, we know about spike timing dependent plasticity. There's lots of different hypotheses for how plasticity could be implemented in the brain. And typically when we want to model these hypotheses using biological neural networks, what we do is we set up either a rate network or a spiking network, which consists of a bunch of presynaptic neurons that feed into a postsynaptic neuron, and they're connected with some synaptic weights. And then we hypothesize an update rule for the synaptic weights. And this update rule is typically in the form of some function that we're hypothesizing. And if you run the network forward, and what I mean by this is you update the postsynaptic activity and then concurrently update the synaptic weights, and you keep doing this, you end up with some activity that you've with some postsynaptic activity trace that you've produced from your rate network. And you typically look for properties of these, whether properties of this activity trace match whatever activity that you've observed. And this is how you sort of validate your synaptic plasticity mechanism that you've hypothesized. what Basile and Tim did about 2 years ago was to say, well, we want to learn these plasticity rules directly from data. So we want to meta learn these rules. And what that meant was basically replacing your functional hypothesis with a parametrized version of the function. So you basically have parameters for this function. And you want to learn the parameters of this function directly from some observed data. And how Basile and Tim did it was to basically minimize a loss function between the activity that came out of the rate of the spiking network and some measured ground truth activity, and use this loss function to update the parameters of the hypothesized synaptic update rule. And so what this scheme was supposed to do was to allow scientists to basically flexibly hypothesize have flexible hypotheses for synaptic update rules without having to impose some functional form on it. So you didn't have to guess at the update rule. You simply had to put in parameters that would allow you to learn what the correct update rule would be from the data directly. And so what this meant was that you could potentially learn a wider variety of rules than simply just hypothesizing a single function. And this is in principle a good idea to do, because then instead of verifying by hand single functional forms for the update rule, what you could be doing instead is to have a whole family of rules that explain a plasticity mechanism. However, the problem is with the loss function. The loss function becomes harder and harder to define. For example, if the number of postsynaptic neurons increases. So if you have a larger population of neurons from which you have observed traces and generated traces, then computing this loss function becomes hard. If you care about certain features of the data, for example, bursting behavior in the traces, then again, defining a loss function for this sort of feature becomes hard. And what do you do then? This is where the GANs come in. So essentially what you would want to do is replace the loss function with a discriminator network, as we have been doing for the previous application. And so how this works is you have an update rule which you've parametrized, and this could be a neural network, this could just be some polynomial with coefficients as parameters. It could be any flexible way of defining a function with parameters. And then you have a discriminator network, which takes the input from the activity traces that you generate by running your network forward with this update rule. And the discriminator would classify this activity as fake. And you have your generated activity, which the discriminator would then classify as real. You train these two networks adversarially, and at convergence, what you should have is a rate network and an update rule that together end up producing activity that looks exactly like the activity that you've observed or measured. And what you will have learned is a synaptic update rule that could plausibly explain the synaptic plasticity mechanism that leads to whatever observed activity that you have. And the nice thing about the setup is that it's completely agnostic to any functional form. You could potentially plug in whatever flexible function approximator you wanted over here and learn it directly from data without having to a priori impose some functional form either on the update rule or on the loss function. And therefore, conceptually, what you could be learning is potentially a family of rules that could explain your observed activity rather than a single functional form. And this is why it could potentially be advantageous to use this setup rather than imposing a particular hypothesis of a mechanism as people have been doing so far. We wanted to test this setup conceptually. And so what we did was we started with Euler's rule, which is it's a Hebbian learning rule. And what we did was our ground truth data was simply simulated activity from a rate network where the synaptic weights were updated with Euler's rule. And this is basically the functional form for Euler's rule. It's the postsynaptic activity multiplied with the presynaptic activity and also multiplied with the synaptic weights from the previous time step. And if we run this network forward, then what we end up is with our ground truth traces that we want to fit our new rules to. And then what we did was for the GAN, we set up several different parametrized update rules. The first update rule that we set up is something that I'm calling the constraint network. And this is basically just Euler's rule, but with the coefficients of the two terms being parameters that we would need to learn. So essentially θ1 should converge to 1 and θ2 should converge to -1. And this is the first network that we're going to try to train, so learn directly from our ground truth data. The second network is just a neural network, specifically a multi-layer perceptron, which is just three layers of artificial neurons stacked with a nonlinearity in between. And this network is completely unconstrained in the sense that it could potentially learn any function to a reasonable degree that is completely flexible. And this multi-layer perceptron or MLP has absolutely no constraints on the kinds of rules that it could learn. So potentially a whole an infinite family of update rules that you could learn with this function. And finally, what we did was to have a biased network, which is just the MLP in addition to Euler's rule. So essentially what we're doing is that we have Euler's rule, which gives us the actual ground truth weight update. And to this, we add whatever comes out of the MLP. And essentially what we're learning here is perturbations to Euler's rule. So what this would do is basically tell us if there are rules that could potentially just be perturbations to Euler's rule that also explain our ground truth data. And we fit all of these networks adversarially. And what this means is that we use the exact same discriminator for all three of our networks. And we basically use the min-max loss to try and match whatever comes out of the rate network with our ground truth data. So when we do this, what we mean is that we have multiple different postsynaptic activity traces from the ground truth data set. This is what they look like. And just to sort of, this is a fairly qualitative plot. And we also plot the autocorrelation of these activity traces here. And then what we have here is a proxy for the trajectory of the weights as you run the network forward. With Euler's rule, we know that the weights should converge to the first principal component of the presynaptic activity, and this is basically what we compute as the weights get updated when you run the rate network forward. When we fit the constraint network, which remember is just Euler's rule with the coefficients parameterized, what we find is that basically we're able to learn exactly Euler's rule. So we produce exactly the same ground truth data. We have exactly the same autocorrelation. We also have exactly the same weight trajectory. And essentially what this plot shows us is that our setup works. You can learn the correct update rule using a GAN setup. And then when we fit the multi-layer perceptron, so just the neural network that's completely unconstrained and could potentially learn any rule, it produces activity that doesn't look very similar to the ground truth data. This is also borne out by the autocorrelation. And also the weight trajectories look completely different from the ground truth weight trajectories. And interestingly, when we fit just perturbations to Euler's rule, now we're producing data that looks or postsynaptic activity that does look like the ground truth, and this is confirmed by the autocorrelation by the autocorrelation plot here. But interestingly, the weight trajectories look completely different from the ground truth. And so what we can infer from this is that the set of rules that we're learning here aren't Euler's rule, but we are producing outputs that look exactly like Euler's rule. And what this means is that there's a whole family of rules, even in something as simple as a case as this is, in a relatively small rate network. There's a whole family of rules that could potentially produce the same output as if you had Euler's rule. And therefore, it's interesting to think about fitting flexible function approximators for learning plasticity rules, because even in this simple case, it seems that there could be a whole family of functions that could explain the same observed activity rather than just a single functional form. We did do one additional analysis where we still fit Euler's rule, but now we made the postsynaptic activity noisy, and we also increased the number of presynaptic neurons. So the details don't matter. Essentially, it's just made the simulations a little bit more complex looking. And here we fit just the MLP, and we find again that it doesn't produce, it neither produces postsynaptic activity that looks similar to the ground truth, nor does it produce wave trajectories that look similar to the ground truth. But then when we have Euler's rule plus MLP, so just the biased network, then we were a lot more likely to find both the ground truth activity and the weight trajectories. And so what this tells us is, in this simple case, at least, when you increase the complexity of the data, then you're more and more likely to have just a single functional form that explains, a single functional form for the synaptic plasticity mechanism that explains the data that you've observed. And so to summarize what I've just talked about is that we aim to meta-learn plasticity rules using GANs, and we tested our setup using Euler's rule. And essentially, the GAN setup is flexible enough both to fit extremely simple setups as well as complicated ones. And in both settings, you can either flexibly learn a whole function of rules, but it also has the capacity to find a single functional form for the plasticity mechanism. But both settings are learned completely from data, and this is why it might be a good idea to learn these rules directly from data using GANs, because you have a lot more flexibility when it comes to hypothesis testing for plasticity mechanisms. And so with that, I want to quickly give you an overview of what other open questions there are for this particular project. One problem, of course, is that we learn these kinds of neural networks that implicitly represent the synaptic update rule, but we have no way of interpreting them because they have no functional form. So this is something that we do need to think about. And also, even if we are able to find a functional form for the trained networks, we also need to think about how we can then infer something about what is happening in the brain from these learned rules. And both of these are questions that we are still thinking about, and it would be good to get feedback for. And with that, I just quickly want to conclude sort of an overall setting for what I've been talking about so far. What I've shown you basically is that GANs are really good models for generating realistic data, and they're particularly useful in settings where the loss function is hard to define and the data is high dimensional. And both of these are cases that turn up quite often in neuroscience, and therefore it's nice to think about using GANs as a solution for these kinds of problems. They're incredibly flexible and are capable of scaling to high dimensions, and therefore could be useful for a potentially wide range of applications. However, one thing that I do want to mention is that they're very hard to train. They're sensitive to initial conditions of networks and hyperparameters, and they're prone to something called mode collapse, which means that the GAN generator just ends up producing the same data point over and over again. And also they could consume a lot of computational resources. So you need GPUs and CPU clusters to be able to train the really big GANs. And therefore, both of these are sorts of stings in the tail when it comes to using GANs. And with that, I want to thank MLS, our group, and our group members, all of whom had a lot of feedback and help, provide a lot of help in both of these projects, as well as some of our external collaborators and funding sources. And thank you all for your attention. And I'm happy to take questions.
Thanks a lot, Purnima. Thanks a lot. That was really, really, really interesting. So to the audience, once again, feel free to ask any question you might have in the chat. For the moment, we don't have any. So I'm just going to go ahead and start. When you specify actually the update rules that you are going to train, so you just try for the moment with an LMP and with OIAS rules.
Yeah.
Did you try more, I would say, biologically plausible learning rule? I think this is an interesting field of research to not only to have computationally relevant learning rules, but also biologically plausible ones. It would be possible to train these kind of rules with your framework?
Yes, in principle, yes. We haven't tried it out because, as I said, the project is pretty in initial stages, so I haven't managed to do experiments that don't involve OIAS rule. But yes, in principle, we started with an MLP precisely because it's completely flexible and we make no assumptions about what the plasticity mechanism could be. But there's nothing in the setup that prevents you from imposing biological constraints on your hypothesis for the rule, right? So I also have to admit I'm new to the field of synaptic plasticity, so I'm not entirely sure what you would mean by a like how you would parameterize a biologically plausible update rule. But you could, for example, impose inductive biases into the network architecture that encode biological constraints, or you could sort of fit the setup with parameterized polynomials or parameterized versions of some other functions, and then nothing about the GAN setup prevents you from making your hypothesis about the synaptic plasticity as flexible or as constrained as you need it to be.
Yeah, I think that would be a very interesting area of research. Trying to train really biologically plausible learning rules, that would be really cool. All right, so we don't have any other question from the audience. Last chance to ask any question you might have. And if we don't have any more question, Purnima, thanks a lot again for the very interesting talk.
Thank you.
And I hope we will see each other in future conferences.
Yes. Yeah. Thanks a lot for having me. I also had a really good time. Thank you.
Thanks a lot for the talk. That was really nice. And you have some thank you from the chat.
Ah, thanks. Yeah.
Awesome. Thanks a lot, Purnima. Thanks a lot to all the attendees, and see you next month for our next NeuroMeetup event. Thanks again.
Yeah, thanks.
Thank you. Bye. Bye, Purnima.